Hey, exoplanetary explorers. Welcome back to In Other Waters with me, Sea Lord Janda. We've just returned from finding the empty dive suit of Minnie. Our user, Ellery Voss, has some sort of possibly romantic connection to her, but we can't be sure yet. We're going to read what we have, their newest diary entry in the crew terminal. Personal log 02. Minnie's suit, open to the ocean, empty. What has she done? I'm trying hard to reconcile the Minnie I once knew with the evidence in front of me. When I met her on Kepler 62F, she was intense and quiet, but calculated, intelligent. She wasn't someone to take on necessary risks, to leap into the unknown. At first, I found her impossibly intimidating. Her experience working as a biologist far outstripped my own. Michael brought me to the site as a consultant to examine her discoveries, but I felt like the one under examination. The base was much like this one, a smaller, older model sunk in a subglacial ocean. Someone might think a relationship was inevitable given those intimate conditions, but it wasn't the base that brought me to her, it was that intensity. Over the few years since, I've tried not to dwell on what happened on Kepler 62F, but being here, living where she's lived, following her scattered steps, it's impossible not to remember. Okay, so Ellery and Minnie did have a relationship. That was highly implied before, but it's certain now. And then we have additional taxonomic information based on a couple of new things we found, plus some... we've got more samples to analyze. The reef cap. Large domed stalks, which, like terrestrial mushrooms release spores from gills on the underside of the cap. In the wider stalk ecology, reef caps appear to play the role of emergency response, growing in areas where the stalk colonies are failing or are heavily predated upon, and proceeding to shower them with spores. These spores seem to differ from those released by other species of stalk in that they quickly dissolve when expelled. Analyzing these is important to better understanding the reef cap's role. Are they reproductive spores, tasked with adding newly seeded stalks to existing colonies? Or nutrient spores, intended to boost the health of dying or deceased individuals? We don't know, but we did see these in areas where they had harsh conditions only. I believe we have a spore. I don't know if we have tissue, but let's see. Oh, I need to unload. Where's it? Here we are. Need to unload all of our samples. Don't forget to bring a couple with, but yeah. Right, back to the lab. We do have stalk spores. Complex cocktail of minerals and compounds. No, that's the wrong spores, apparently, but that tells us more about the reef stalk. Analysis of stalk spores has revealed a rich cocktail of compounds unique to each spore. No, we read about these already, didn't we? I'm sure we did, yeah. Why was this labeled as new? I honestly thought we had... these. Yeah, this is the one. Okay. Soothe spores. Yeah, reef cap taxonomy. Okay, that's completed now. The reef cap, Collis agaricus. Soothe spores, the name by which I've come to call the reef cap spores, don't seem to be spores at all. Unlike the modified spores of other stalks, these particles don't have any reproductive capacity. Instead, they resemble endospores, dormant bacteria reduced to dried husks which can survive for hundreds of years in toxic environments without nutrients. Endospores are incredibly tough and high in calcium. When released by the reef caps, they dissolve and transfer this calcium to the surrounding environment, where it's absorbed by the local stalks. This windfall of calcium, granted by the cap's soothe spores, seems to allow stalks to augment their internal signaling and toughen their membrane to protect from predators, allowing them to recover rapidly. But the question remains, how do the reef caps produce these endospores? Okay, so they are deliberately aiding the other local spores, which all of this plays strongly into the idea that this is a very complex and at least pseudo-intelligent ecosystem. Like, the reefs, everything, all the reef stalks are connected somehow. Then we also found the table stalk, Collis canopium. Pale yellow canopies formed from many stalks entwined together, which reach impressive heights. Table stalks are part of the reef's many interlocking species of stalks, although these individuals can only be found in the eastern reef. There they tower over the other creatures, providing dappled shade for the life that grows around them. One of the notable features of table stalks is the rhythmic opening and closing of their pores, which provides a steady flow of water beneath their canopies. This seems to be highly beneficial to the growth of other species, with most table stalks being surrounded by many types of stalks and other species entirely. Hold on, I'm trying to... Okay, so they deliberately have water flow through them, which helps other species. Also notable are the ball-shaped growths of differing sizes which sit high in the table stalk's canopies. Sampling these might help us understand the table stalks better. Right. Well, we do have more samples. I should probably just analyze all of these. I feel like we analyzed both of these before. But anyway, here's the stalk roots. Tangle of silken roots from a stalk colony. Oh no, that's the other... Oh, okay. Well, canopy root is here. Root of a stalk canopy. Many twisted filaments. I'm just going to analyze all of these real quick. This is the pollen from the single petal creatures, and... Oh. This is a bit of tissue from one of the bubble stalks. All of these are useless now. These, I think, are not erased because they're actually useful to us still. Well, that's not entirely true. The shrill sacs and such are. Not totally sure. Anyway. That did give us the last information about the reef stalk. Okay. Theories. Close analysis of stalk root networks reveals something surprising. The root networks are in fact part of a symbiote, a different fungal species entirely which exists within the stalk's bodies. This fungal symbiote provides a carbon exchange network, which runs throughout the bedrock of the reef, connecting all colonies in a single web. This symbiote also produces the spores which the stalks use to communicate, but these spores are edited by the stalks themselves to act as reproductive agents, information carriers, and chemical delivery systems. If the stalks are able to edit the genetic signatures of these spores and their parent species, their intelligence and complexity must be way beyond what I previously theorized. Close analysis of stalk spore language could reveal an entire corpus of communication. Are there stalk jokes, stalk histories, even stalk literatures? Okay, so they're a network and they're very intelligent, actually. And here we have a sketch of our first sketch of what a reef stalk actually looks like. I mean, it's pretty much how I would picture it, but yeah, there it is for anybody who's wondering. It's got pores, it's got chitin plates, little growths. It's a stalk of fungus. Right, and we also got the last thing for signal stalks. Chemical analysis of a signal stalk tissue sample has confirmed the presence of a yeast-like fungus and large amounts of ethanol saturated in the tissue itself. This prompts a rethink of why many creatures target the hard-to-get signal stalks as a foodstuff. While I initially theorized it was due to their role as protectors of stalk colonies, the significant presence of ethanol in signal stalk tissue means most creatures would feel the effects of the alcohol consumed and make it buzzed from grazing on signal stalks. This in moderation would be a favorable, even enjoyable effect for the creatures, leading them to repeatedly target signal stalks. If I remain stranded on Gliese 67cc for long enough, I may need to make use of this particular biological property myself. So the signal stalks, they provide the warnings for the colonies, but apparently they also produce a bit of yeast and ethanol, and so they can make the creatures drunk. But yeah, they're basically extra tall stalks covered in these sacs which explode and can be heard, or maybe the vibrations felt by other stalks as a warning system. It's an interesting sketch. So there's our sketch of that. And then the table stalk behavior. Analysis of a table stalk root shows a number of minerals not associated with stalk growth being extracted from the bedrock. Do the table stalks need these minerals to survive, or are they using them for some other purpose? A cross-section of the root shows a central chamber typical of the carbon transfer system seen in other stalks, but unlike those stalks, table stalks have many subsidiary chambers, like a bundle of wires, each carrying a particular cocktail of mineral compounds. These secondary roots are not drawing up large amounts of these materials, just traces. But why? So they're pulling random minerals out of the bedrock. Perhaps there's some connection to those growths in the upper canopies of the table stalks. They are well secured by the stalk's tight binding, but perhaps we can find one that's been dislodged. Perhaps we can. And then we got our first information on Plantae novi, punctifolia, the pore petals, shimmer blooms, or Coltor minae. Shimmer blooms are single-petaled plants found primarily in the sheltered waters of the East Reef. As they produce pollen as well as photosynthesize, I've classified the visible portion of these plants as petals, though they could also be thought of as leaves, serving both functions simultaneously. Shimmer blooms can be green, like most terrestrial plants, however they can also be found to be yellow, red, or purple in color. The image of a patch of blooms is a beautiful one, and as they shift in the soft currents, they often seem to shimmer. The lower part of their single petal features many small stamens, which produce a fluffy, clumped pollen. Analysis of this pollen may provide some insight into their life cycle. We got the pollen quite easily. Laboratory analysis of shimmer bloom pollen reveals it to be a heavy, sticky substance, atypical for waterborne distribution. Unlike on terrestrial plants like seagrass, whose pollen is distributed by water currents and is therefore long and light, shimmer bloom's spherical, heavy pollen seems intended to resist distribution by current, and must instead be distributed by creatures who visit the blooms. The reason for this may be the shimmer bloom's tendency to grow on exposed shelves or pillars, which would risk lightweight, water-transported pollen being sucked into the deep ocean rather than being distributed to viable sites. By relying on creature transport, bloom pollen has a better chance of reaching another sunlit shelf or pillar. Yeah, that makes good sense. Okay, so time to go, I guess. Let me grab a couple of, I think this is what we're gonna use as fuel, so I'll take a couple of those. A couple of shrill sacks. You know, I'll just take one. I think we'll have more. One of these. Yeah, those are the actually usable tools. Right, so what are our sort of goals at the moment? Right, we have to go into the West Reef and find fuel. I know we need that to get here. We might need it to get here too. I think we just gotta go west, which we approached once already. Passed down this path before. This is maybe the ruins of another substation. And here we are, the edges of the West Forest. Mayonnaise map data mentions a microbe which only occurs in this stock forest. If we can find 4 healthy colonies, it'll make the ideal basis for the propulsion system's fuel cell. Okay, well, there it is. They're in curving rows, tightly packed and evenly spaced. So we're going to need to use a shrill sac to blow that open. Yep. Well, here we are, a new plant. A bloated stalk. These stalks bulge out from their bases, forming rounded, pulsing growths that look discolored and stretched. And Ellery is logging data on them. They look ill. This growth is bloated and distended, the panels of chitin bulging outwards. Between the seams, something flows within. Well, the microbe we need for fuel, perhaps? What are those strange inflated stalks? They look like the sporing growths we found in the eastern caves, but there's something wrong with them. We need to break them open somehow so we can sample the microbial colony within. See if they respond to the shrill sacs. Well, I already detonated one right next to this one, didn't I? No, I guess not. Okay, well, I have another one, I think. Oh, yes, okay, we're in a pool of microbes now. That's fine. They're only alien microbes. 30% microbial. And we've acquired a spark colony. We've got one. The clouds of microbes these stalks release are incredibly toxic. That's great. The suit is having to flush its vents with oxygen to protect me. We've lost a lot of reserve oxygen. That's nice. We're going to need to be fast when sampling these bloats. Let's get another 3 colonies. But be careful. I don't want to run out of oxygen out here. Yep, makes sense. So we should not hang around. Now here's a glittering stalk. We need more shrill sacs, clearly. To the west, the stalks spread in thick pathways, curving between strangely corroded stones. As they curve south, the stalks meet the rising wall of rock which shelters the southern reef. Okay, let's go south. That's more branching. Alright, is this another bloater? Yes. The membrane of these stalks is thin enough to be translucent in areas, stretched to breaking point by whatever's inside. The rock behind this growth looks as if it's been melted. Its layers whirl in distorted forms, revealing a metallic core. I mean, it could just be evidence of volcanic activity or something. Grab ourselves some spark colonies. There's only one here? I guess so. Okay, let's move on before we run out of oxygen. Here's another glittering stalk. We're gonna need a lot of shrill sacs to get through this forest, yeah. Fortunately, they seem to be plentifully available. The stalks seem to be growing around the bloated growths, isolating them from each other. What purpose does this serve? Okay, I feel like west is the main way to go, but there was a way to go east, too. Between the rocks and the stalks, a thin, sandy passage leads towards a clearing. A small clearing between the avenues of stalks. To the west, their ordered rows dissolve into the chaos of the southern forest. Now that. I've spotted smaller, spotted variants of these agile creatures. Another species? Or perhaps sexual dimorphism. These might be the males. Ooh, a clouded shell. Fragments of a shell. Recently shed, glint in the sand like shards of cloudy glass. What creature left these here? Well, I would assume it's one of those living creatures. I mean, the animalia, as it were. 10% animal, 0% shell. Piece of a creature's shell with a cloud pattern. Okay, yeah, so it was worth taking this little detour. We got a critical sample for studying the animals. We haven't managed to click on enough to actually unlock study of the animals yet, but we will pretty shortly, I think. West, and here we have another bloated one. The stalks form a loose perimeter around this growth, which bulges unsettlingly as if it's about to burst. Okay, there must be some sort of... These microbes must be some kind of disease or something. The other stalks don't seem to like this at all. When alarmed, these stalks burst open, releasing their toxic contents into the local water. Could they be defending themselves? Maybe, but it doesn't seem the other stalks appreciate it if so. Spark colony grabbed. Let's move to... Be easy to become lost in the complex patterns traced by these rows of stalks. I wonder where they lead. Still not enough oxygen here. Gotta move. This stalk sits on top of the corroded husk of a boulder, melted down to little more than a pebble. These seem heavily corroded. I wonder if these oh, full. Crikey. Well, I've got enough shrill sacks. I mean, we'll just keep going, I think. I wonder if these bloated things melt them. To the west stands the final pathway of stalks before the colony fades out into the sand and rock. There's something different about the growth patterns in this forest. The ones further south were more chaotic, but these stalks are almost arranged avenues or terraces. Do you think they might be protecting something? I almost feel more like they're fighting something, but maybe. What would they be fortifying? Why all the diseased bloats? The pulsing, pumping rhythm of these stalks' contractions suggests that some process is happening inside them. Well, probably producing more microbes, but these bloats pulse unnervingly, as if at any moment they might burst open, choking the water with a microbial cloud. Seems like an accurate observation from all we've seen. Great, we've got plenty of these microbial colonies, but where did this infection come from? The stalks seem to be struggling to deal with it. Or is this just part of their expected life cycle? What do you think? No, I think this is abnormal. I agree, this feels like a rare event, an epidemic perhaps, but what's the source? Either way, let's bring these back to the base as soon as we can. We can analyze these samples there. Call in the pickup. Not yet, but also maybe let's walk away from this. This is the western edge, there's a rock wall. Let's try to get out of where we're dying due to the lack of oxygen real fast. Oh, wait. Go further west. The western wall rises up towards the shallowest part of the shelf, just meters below the surface. So the reef is just barely... Oh, what's this now? Below the steep wall, a dark, crescent-shaped sinkhole cuts into the shelf. Fallen rocks from the wall are strewn across the sand. Secret cave. Into it at once. It's not big. There's probably something down here. Whorled passage. The walls of the passage twist inwards with thick veins of growth. We must be heading beneath the stalks. Ah, thick, bloated roots poke through the rock wall and something pulses within them. A rare chance to sample the stalk root network. And it must be of one of the bloated ones, yeah. Of course I feel like maybe I was wrong about this stalk bark. We'll connect up. I need 4 of these apparently. But the soothe spore is proved pretty unhelpful in this area. Let's dump that. %, fungal, 20% root. We should only need one sample. It's a bloat root, yep. Alright, so we got the secret that's in this little cave. No way to go south, so up we go. We are a touch low on oxygen, but I think we're fine to keep wandering around here a little. There might be more. Oh. Okay, let's hit him with the shrill sack. Oops. Great. We're gonna have to hit him with another shrill sack, because I put that one in a bad place. This side of the colony loops back towards its center, leading deeper between the stalks. That's fine. This is probably a loop, but we'll follow it. Another bloated growth. Some vein or root must run through this spot as the sand rumbles with unseen movements. Are all the bloats connected? There we are. Reef bloat is what we're calling it. Enough data to log and classify these stalks. We're calling them reef bloats, yeah. Large, bloated stalks that burst easily, expelling the toxic microbes held inside their bodies. Their movements suggest unseen processes. The bloated stalks seem to be precisely spaced out between the stalks. Are we damaging this colony by disturbing them? To be honest, that is a possibility. Seems unlikely the bloats are simply individually diseased stalks. Their growth patterns suggest a system, a process. They are quite organized. Give them that much. I need more shrill sacks. Yes, we're kind of critically low on oxygen. But it's fine. If we get really close to running out, I can just call. Maybe I was wrong about this being fuel. I'm just going to dump it. We can call the thing in at any time, so if we get down to 0 oxygen, I'll just do it. I was hoping to get both, but I think I've actually gotten neither. That's unfortunate. What's up here? Another bloated growth. With its chalky covering melted away by the bloat, this rock has a dull metallic sheen, spectrums of light reflecting across its surface. Don't need any more samples, to be fair. There's a sheer ridge rising up to the north. North of here, an angular ridge ascends, cutting the shelf in two. The only way further north is to cross the rift to the east of here. Ooh, hollow stones. Tucked into the side of this corroded rock, a pile of stones with worn holes sit in the sand. Within a hole, something glitters. And I startled another one of those agile creatures. Got it! Agile, jet-propelled creatures with colorful mantles, found hidden amongst reef stalk colonies feeding on their spores. So we're calling these spore catchers. The fast ones. Fair enough. Let's sample what we've got here. Question mark, 40% animal, 20% egg. I wonder if we've got a spore catcher egg here. I should probably only need one, but I'll grab two. And here's another glittering stalk. Good, I need this. Behind this tall stalk, layers of thick growth surround a maze of rocks, blocking the passage back east. Striped egg, we're calling this. Yeah, okay, so it's definitely an egg. I'm gonna drop one. I think we should only need one. Because I need more drill sacks. Okay, yeah, this was another dead end area. Good news is I think we've explored thoroughly enough that there's probably not anything left in this reef area. But we'll find out, I guess. Bubble-skinned, glittering stalks keep their distance from the bloats as if they know the danger they pose to the stalk colonies. I wonder if they're the stalk equivalent of, like, landmines or something, you know? Or they are just diseased, it's hard to be sure. Clearing these stalks is a delicate task, and I hope their retraction is temporary and not some terminal state. Well, I'm fairly sure it's temporary. We are kind of screwing around with things we don't really understand, though. Very human of us. Another bloated growth. It's in a strangely shaped clearing. We still don't know why they grow like this around those. A pulse can be felt running through the shelf from the north, making the sand ripple in small, soft waves. That went back to a place we'd already explored, I think. Right? Well, I haven't clicked on this, though. These stalks are isolating the bloats. Perhaps it's because they're dangerous to the colony itself. I've been there, though, yeah, so... Back this way... These curving avenues of stalks seem to mark a loose perimeter around each bloated growth. Why? Why indeed? Oh, and we've been to this. This is the other edge. Alright. I think that's probably everything now. Let's leave the Western Reef. Retrieval requested. Resyncing. Booted. Dive map offline. I guess I gotta talk to Ellery. Hey, you ready to get back to work? Yep. Great, because I've had a very productive few hours. Excellent. I've been working on that propulsion system. I've used those 4 microbial samples to grow a new colony in the lab. Then I transferred it to a secure containment capsule, nice and snug. I've followed what I could from Mine's notes on building a microbial fuel cell. It's all loaded onto the suit, the MFC fed directly from our reserve oxygen supply. That sounds like our reserve oxygen supply might be broken now, but okay. Now we just need to take the suit out for a dive to test it. It's going to take plenty of oxygen to fuel the colony, so bring any samples that'll help out. But make sure not to take any we haven't put through analysis on the lab level first. I don't want to burn through hours of research material just to get a power boost. This propulsion system will let us pass through strong currents and move more quickly. We should be able to cut across that rift north of the way station and see what the bloom finally is. Ready when you are. Okay. Let's read there. We read this already, didn't we? Yeah. I don't know why it said it was updated. I don't think it is. Dive map. Oh yeah, the next place, story-wise, to go is the north, but I think with this power boost we should be able to pick up all of these extra samples while we're at it. Lab-wise... well, okay. Fungi, Colodia, the reef bloat, Collis tumidus. Reef bloats are large, bloated stalks that can be found at the western border of the reef. Intimidating in their size and distorted shape, they resemble inflated, sporing mushrooms, covered in nodules of rampant fungal growth. Their membrane, which is stretched by the bloating process to an almost translucent sheen, is darker than a typical stalk, making the stalks themselves a shade of sickly orange. What role the bloats play in the reef's stalk network is not immediately obvious, but their pulsing, pumping motion suggests that the bloats are either processing or producing the toxic microbes that are contained within their fragile membranes. Without analyzing the microbial colonies themselves, this will be difficult to ascertain. Yeah, so, well, that's true. It could be that they're processing them. That's true. Let's be fair to these reef bloats. We're prejudging them a little. They could be extracting these from the water and processing them. That's a possibility. And then Animalia novi, Myomecia, soft bodies. The spore catcher, Legulus striatus. Spore catchers are small, soft-bodied creatures which move quickly through the water by using siphon jets. Their mantles, striped with bright colors that shift as they swim, allow the creatures to modify their movement through the water with precise flapping motions. Watching them navigate the water is hypnotic, their rapid turns and loops showing incredible agility, flitting through the water like a bat or bird. Spore catchers use this wide range of movement to pursue the various fungal spores produced by the reef's stalks. The spore catchers seem to be highly selective about which spores they wish to consume, suggesting that analysis of the stalk spores might tell us even more about the spore catchers' behavior and diet. Oh, so we have to analyze their food in order to... Okay, I think we have all the samples we need of that then, probably. As soon as I unload our suit. These are actually good for samples. These are spores, they were food. That's the last thing we needed those for scientifically. Okay. Do we have any? Apparently we don't have, we're not out of uses for this, but... taxonomy update. I don't think it actually is though. I think probably we had that already. Let's analyze these spark colonies. Obviously that'll tell us more about the reef bloat. And that's the only use we had for those for these purposes. And the bloat roots, so that should be everything on the reef bloat, I think. Striped eggs. Spore-lined nest, that's interesting actually. That should complete the spore catcher thing. Clouded shell. This is for the other animal. We didn't actually discover the other animal fully yet, yeah. We just have to click on a couple more. They should be around somewhere. Okay, Calodia, reef stock. Oh, we did get the last thing filled out of this. No, we already did. Yeah, we did. I don't know why. Well, just it updates every time I analyze. It's a relatively primitive game in that regard. I knew this about these two. I just analyzed the stock mark a second time. This is new, though. Laboratory analysis of the microbial colonies found within reef bloats, spark colonies, as I've come to call them, shows that these microbes are not of a group typically associated with stalk colonies. In fact, spark colonies appear to be made up of highly modified algae, with a wide metabolic range, able to consume carbon dioxide, oxygen, metal, and toxic compounds with equal ferocity when supplied with a source of light. When metabolizing these substances, spark colonies produce a regular flow of charged electrons, making them ideal for use within a microbial fuel cell, or MFC. Their suitability for this use is suspicious. Are these really naturally occurring microbes? And why are they found only within reef bloats? Something isn't right here. Yeah, that is awfully convenient. Theories. Finding a root sample from a reef bloat has brought with it the confirmation that reef bloats are not manufacturing spark colonies, but instead modifying, metabolizing, and processing them. Analysis of the bloat root system has shown the presence of microbes at various stages of modification, including those that had been entirely detoxified and their rampant metabolism neutered. If all reef bloats are connected via this root system, then they may be part of a large-scale cleanup operation with the aim of eradicating these aggressive microbes. Reef bloats are not diseased stalks, then, but powerful biological engines fueled by their local stalk colonies. The scale of this operation suggests that for the stalks, the spark microbes are a significant threat. Okay, so these are like filters. They're cleaning up the microbes, not producing them. Good to know. Oh yeah, that's real bloated though, huh? Bulging, discolored. No, they're kind of suffering from this process of cleaning them up then, I guess. And breaking them open is probably not winning us any favors among the reefs, huh? Sorry, guys. Or among the stalks. Sorry, guys. As for the spore catcher, behavior. Analysis of the unique chemical compounds found in stalk spores and a wider understanding of how stalks use spores to communicate suggests a more mutual relationship between spore catchers and stalks than initially assumed. If stalks are able to tailor the chemical makeup of their spores, it's possible that the select spores the catchers eat are intended for their consumption and contain compounds and minerals attractive to the species. As spore catchers use their color-shifting properties to hide among stalk colonies, it seems increasingly possible that stalk colonies and spore catchers are mutualistically linked, and catchers serve the needs of specific stalk patches. What those needs are, however, is not yet clear. Analysis of a spore catcher egg, if found, may help us gain a full picture of their physiology. Well, we found one. Congratulations, Dr. Foss. Analysis of the spore catcher egg shows a strong correlation between the minerals it contains and the minerals present within the catcher's favored spores. It seems that through the spores, stalks are able to affect the catcher's egg production, therefore controlling the volume, traits, and behavior of catchers within any area of the reef. In a sense, the stalks have domesticated the catchers, leaving the two intrinsically linked as species. But the question of what the catchers provide the stalks remains. Do catchers serve a specific purpose, consuming unwanted or incorrectly configured spores, for example? Or are they kept for other reasons? Could the catchers be kept by the stalks for their companionship and social use, just as humans keep pets? That would be fascinating. We have uncovered a lot of the evidence we've uncovered suggests that the stalks are really pretty damn smart. But would they have a consciousness capable of that sort of thing? Maybe. They're kind of cute looking. They got flaps. If they're colorful, I believe, right? Yeah, they're very colorful. They move quickly about. They're almost like underwater butterflies or something, sort of, or dragonflies maybe would be more apt since they seem predatory, but, well, I guess they are herbivores though, really. Anyway, I believe that's all the samples we've got, well, except this one, but we don't know what to use that for. What would have oxygen in it? I still feel like it was this. But probably bring the spark colonies too, at least a couple of them. Maybe we can pretty much feed anything into this engine though, actually. Alright. So, we need to go this way to advance the story, but we're gonna head out this way first. That's gonna do it for this episode though, guys. Thank you everybody very much for watching. And I hope you all have a nice day, crew.